So this is the 5th respiratory physiology, and it is about the carbon dioxide dissociation curve. Again, we'll run through concepts here that have been developed in the previous lectures. Please watch them in order. We'll try to demonstrate what the questions tend to ask for your Viva, go through some background knowledge, and there's a few important distinctions to make in this particular chapter because of terminology more than anything. I think everyone has seen the factors that go into moving carbon dioxide. It's just some tuning up of the terminology that we'll need to come to grips with. And once we've been through some background knowledge, we'll go to our model answer. The questions that are asked in this particular section are very similar. They don't vary a great deal. There's not much depth at all associated with them. Like I said, it's mainly about the terminology that people come undone. They will relate mostly to the curve, so the diagram that we're going to construct. And you should be able to answer most of these by talking to that diagram. And they also relate to various eponymous parts of that diagram. So the Haldane effect, for example. We'll run through that. The ways that the questions are asked, for example, in what form is carbon dioxide transported in the blood? Please draw the carbon dioxide dissociation curve for normal arterial blood. Or draw a graph showing the relationship between the pressure of carbon dioxide and the total carbon dioxide content in blood. Where will the curve lie for venous blood in relation to arterial blood? Or does the curve move up and down between arterial and venous blood? How is carbon dioxide transported, and does venous blood carry more than arterial blood? Or how does venous blood do this? What is chloride shift? What is the Haldane effect? And so these are all fairly straightforward ways of asking similar things. There's a body of knowledge here that we'll try and get through. I think the first place to start is with our blood that we spoke of before, that we started to build. Recall that we had a vat of blood. You had an air-liquid interface, and across there moved gas in proportion to its partial pressure. And this was Henry's law that stated, in this case, we're going to have not PO2, but PCO2. And it will move proportionally into the blood across its membrane. And when we do that, there will be an increase of the PO2 of dissolved gas, an increase in content, therefore, of the gas, and it's in proportion to the PO2. And that amount will increase as the external CO2 increases. So if we were to relate this to an alveolus, we'd have alveolar CO2 resulting in an increase in blood CO2. And that would be purely through dissolving a gas in a liquid medium. Now, the amount of carbon dioxide that can be dissolved in that way is not very much, proportionate to the body's requirement to shift carbon dioxide. In fact, this amounts to only about 7% of the requirements of normal metabolism. So this alone is inadequate. It is possible to allow increasing partial pressures of CO2, and that would increase the proportion that's moved, but CO2 also participates in acid-base balance, and that seems to be prioritised over the CO2 or PCO2 content of blood. So it is very tightly regulated and not much room to move. Therefore, another mechanism is required. Just as we spoke previously of compartments in our blood, we're going to create a CO2 reservoir. And we're going to allow CO2 that is dissolved in the liquid to enter our little reservoir that we make, and that will remove it from the plasma component of blood, allowing transition of more CO2 into the blood. But that particular CO2 we're also going to package into our reservoir, and so on, resulting in an influx of CO2 from the atmosphere across the membrane into solution and then packing away into a reservoir. And what that does is facilitate, using this reservoir, a whole lot more weight of carbon dioxide going into our blood. So the content of carbon dioxide in the blood is far greater if we create this reservoir. We'll talk more about what this reservoir actually is in a moment, but note that it is now capable of carrying the remaining 93% of carbon dioxide that is shunted around in the blood. So the overall content of carbon dioxide increases dramatically because of this reservoir. Now, we developed our blood before by saying that we had red cells sitting inside. And those red cells had hemoglobin in them. And the hemoglobin was able to participate in oxygen transport, and in fact it is the very molecule that has allowed us to exist on this planet. It hangs on to oxygen and delivers its oxygen in under the right conditions to the tissues, resulting in a hemoglobin molecule that has empty binding sites. When those binding sites are empty, that results in a conformational change to the hemoglobin molecule, which changes its affinity to other types of chemicals. Now, it's convenient to say that, well, carbon dioxide can jump on there, but that's not how it works. Hemoglobin is a large molecule, a large protein made up of lots of compounds, and carbon dioxide can attach itself to those compounds after the conformational change. But it doesn't actually attach to those sites. Likewise, it's a large protein. There's lots of compounds within that protein, and that protein, once it's had its conformational change, can bind something like hydrogen ions, but not necessarily on the oxygen binding sites. So what we end up with is a situation in our blood where when oxygen is offloaded, hemoglobin is capable of absorbing some carbon dioxide. Hemoglobin is capable of absorbing carbon dioxide. And so carbon dioxide moves out of solution onto the hemoglobin, and our reservoir starts to fill. The red cell has hemoglobin that becomes filled with carbon dioxide. And this represents one of the mechanisms by which our compartment that we designed in the blood is able to package away carbon dioxide. Proportionately, the amount of carbon dioxide that attaches to the hemoglobin is not this entire 93% of the carriage. It's around about 20 to 30-ish% attaches in that way. And that is convenient. There's a large amount of hemoglobin in the blood, a large mass capable of picking up a large mass of CO2. So that carries a large volume, but is not the biggest contributor. What we're going to do is consider our blood, again, slightly separate situation. We have a PCO2 above it, puts CO2 into the fluid component or the plasma, and the plasma also contains water. Water combines with CO2 to produce bicarbonate. or dihydrogen carbonate, H2CO3, and it's in an equilibrium. And that equilibrium moves carbon dioxide out of solution, allowing more CO2 to enter. Eventually what will happen though is the amount of dihydrogen carbonate will increase, pushing the equilibrium point back so that not much more CO2 can move. And therefore, not much more CO2 will move across the membrane. So that will saturate eventually. But not before this system is able to make up the remainder of this 93%. So what we end up with is about 65% of our carriage of carbon dioxide in the blood is the result of combination with water. There's a few stipulations to put on that to make this a reasonable suggestion. What we observe experimentally is that the rate of this reaction, being in equilibrium, dihydrogen carbonate, this rate of reaction is very slow. And in fact, it's so slow that it can't account for this much carriage of carbon dioxide. So there's something else going on. And what does go on is the enzyme carbonic anhydrase. Carbonic anhydrase lives inside red cells in very high concentration. And what that does is increase the rate of reaction many thousand-fold, such that carbon dioxide coming in contact with water in the right environment with carbonic anhydrase produces dihydrogen carbonate. So that means that in red cells, the carbon dioxide that moves into the red cell, and I'll just expand up our red cell here. We know that hemoglobin's in here. It's 4 little bits. The carbon dioxide that enters our red cell combines with water and using carbonic anhydrase quickly equilibrates dihydrogen carbonate. And so the CO2 has moved out of solution, out of plasma, into the red cell, where it's rapidly converted to hydrogen carbonate. And that then consumes the CO2 that was in here, allowing more to move. And if we allow more CO2 to move into the red cell, we end up allowing more to move into the plasma stage. So what we're doing here is creating, through the use of carbonic anhydrase packaged up into red cells, our reservoir to hold carbon dioxide. But that's not the end of the story yet. Eventually this will saturate. And we also know that this is in equilibrium with bicarbonate ions and hydrogen ions. This is an acid. What we have then is the situation where bicarbonate ions become another large reservoir for CO2 to be stored in. But again, acid-base plays a role, and acid-base is regulated very tightly in physiological systems. So the advantage we have is that hydrogen ions will attach to the hemoglobin, not at particular binding sites, just in the conformed molecule in the deoxygenated state. Hydrogen ions can be picked up by the hemoglobin, allowing the equilibrium point to shift and create more hydrogen ions. But eventually, acid-base will take over. We'll have too much bicarbonate ion. So what we need to do is shuttle bicarbonate ion. We take bicarbonate ion out of the red cell at this stage and puts it into the far greater reservoir called the plasma, the extracellular volume. And so dropping the bicarbonate concentration inside the red cell allows this equilibrium point to shift further. So we also need to understand that moving carbon dioxide from this point into the plasma, into the red cell, into dihydrogen carbonate, into a bicarbonate ion, and back out is the flux that occurs. And it also creates another problem to be solved. We have negative particles moving out of the interior of the red cell. That needs to be balanced. And that's balanced by the inward shift of chloride in environments where there is high CO2. And this process is called the chloride shift. So the chloride shift is the phenomenon that's observed in venous blood that increases the concentration of chloride in the intracellular environment of red cells because bicarbonate has been pushed down. This chloride shift is innate and necessary for the carriage of carbon dioxide inside the red cell. It's innate for the production of bicarbonate ions from dihydrogen carbonate. It's innate because we have presence of carbonic anhydrase, which is able to consume any carbon dioxide that makes it in contact with water inside the red cell. So what we have now is a situation whereby our reservoir is contributed to by two factors, and hemoglobin is integral in both of them. The hemoglobin that sits in here becomes deoxygenated and also contains within the red cell carbonic anhydrase. This deoxygenated hemoglobin is able to accept the hydrogen ions and also able to accept directly carbon dioxide. And that accounts for the 93% of carriage volume of carbon dioxide within the blood. Now, the reverse situation will occur once we get oxygen reattached to the hemoglobin. So if you can imagine this process occurring in areas of low oxygen tension, i.e. in the tissues, in the venous side of the circulation, carbon dioxide is loaded into the blood. We return now the blood to a situation of high oxygen concentrations, the lung. What's going to happen is this process will reverse. The process will reverse so that higher carbon dioxide in the plasma flows along its concentration gradient into a relatively lower concentration environment of the alveolus. If this carbon dioxide is removed, this carbon dioxide will move back out into the plasma. If this carbon dioxide is removed, carbonic anhydrase facilitates dihydrogen carbonate consumption. Dihydrogen carbonate consumption results in consumption of bicarbonate ions. And oxygen attachment to the hemoglobin liberates CO2 and liberates the hydrogen ions. Those hydrogen ions are free to combine down here again. And so we facilitate the movement of bicarbonate back into the cell. We reverse our chloride shift by facilitating movement of bicarbonate back into the cell. And so our chloride shift reverses once we get to the arterial side of the circulation. And that allows the transition of carbon dioxide through and out into the alveolus along the concentration gradient the whole way. Now this is a very rapid process because of carbonic anhydrase. This function of oxygenated hemoglobin liberating carbon dioxide is part of what is known as the Bohr effect. The opposite of that, or the ability of our hemoglobin that doesn't have oxygen attached, deoxygenated hemoglobin, to hang on to carbon dioxide, is what's known as the Haldane effect. So the Haldane effect is oxygen depleted hemoglobin being able to accept CO2. It's not particularly on those binding sites, but the conformationally changed hemoglobin molecule is able to accept the CO2. So, we built our blood, realised that we couldn't dissolve much CO2, created a reservoir and bound the rest of it up. In fact, we bound it to deoxygenated haemoglobin and used the availability of carbonic anhydrase in red cells to convert carbon dioxide into bicarbonate, and used surplus chloride to shift that out of the red cell, allowing more reaction to occur inside. When we had deoxygenated haemoglobin, we're also able to add carbon dioxide to it, and that's the Haldane effect. And we're also able to make use of the property of deoxygenated haemoglobin to be a proton acceptor, i.e. bind up protons, reduce the acidity, and therefore allow more bicarbonate to be produced, or more CO2 to be channeled through the red cells. So what this means is carbon dioxide transport is closely related to red cells. Without the red cells, we don't have the ability to transport near as much carbon dioxide. And so that is the reasoning behind all of the questions we're asked about carbon dioxide transport. Now, there's a bit of nomenclature, a bit of terminology that we have to get through here. Haldane effect is deoxygenated haemoglobin accepting carbon dioxide and giving it up when it is oxygenated again. Chloride shift just keeps electrical neutrality in the venous side of the circulation when bicarbonate is ejected back into the plasma. We talk about dissociation curve now, and everyone's very familiar with the oxygen dissociation curve, and that's very much tied to haemoglobin. Haemoglobin here participates and provides a reservoir for the total amount of carbon dioxide sitting in the blood, but don't make the mistake of calling this the carbon dioxide haemoglobin dissociation curve, because it's not. That's not what we're talking about. The misleading thing here is the dissociation curve for carbon dioxide has haemoglobin participate in it, but is not as big a contributor as it is in the oxygen haemoglobin dissociation curve. What we're talking about for this particular curve is the relationship of total CO2 in the blood, all of this, the actual mass that's available, in proportion to the partial pressure of CO2 that sits in the blood. Now, you'll recall that regulated CO2 is a priority because changes beyond a very tightly regulated system result in acid-base changes, and acid-base is defended far more readily in physiological systems than is just plain CO2. So we end up now talking about the total CO2 in proportion to a very tightly regulated section of partial pressure CO2. Now, these curves exist in different ways. If we extend this axis right out, we end up with these really big curves that look similar to this. We're only talking about the physiological range, between 40 and 50. This little bit here. So I've blown that up for explanation's sake. And what we're talking about here is a curve that looks like this. Note that it's almost straight. There's very much proportionate relationship here. And it's because of all of this that we've been through before. So if we talk about a normal arterial CO2 of 40, we come out with an amount of CO2. But if we talk about a venous, let's be generous, we come out with another amount of CO2. And what we're talking about is the total. And so proportionally what we see here is for a small change in PCO2, such as occurs between the arterial and venous circulations, there's a large change in the total CO2. i.e., the unity curve here is like this. So what that means is we've got a curve that has a steep slope. So the ability of the lung to offload a lot of CO2 is good because of the steepness of that curve. Now, that is the dissociation curve for carbon dioxide. All it does is tell us about the change in the total amount of CO2, the mass of CO2 that sits in the blood, induced by changes in the PCO2, changes in the partial pressure. And remember, partial pressure we can measure with a test tube over here, seal it off, allow everything to equilibrate, and measure the CO2 there. Again, flowing on from before. All we're doing is relating this bit of CO2 that's able to move between states and become measurable to the overall content that sits inside the blood. So if we relate just a small change in that PCO2 to a large change in the total CO2, that's what we're talking about. I'm really trying to emphasise this. Likewise, we try to emphasise the total oxygen content in the haemoglobin dissociation curve. Once you get your head around that concept that we're talking about a mass, an amount, then we can progress the thinking. So we now have the situation whereby on a given curve, this proportion is always the same. What we can do is make use of the Haldane effect. Recall the Haldane effect was the one that changed the ability of haemoglobin to bind carbon dioxide when the oxygen binding sites of the haemoglobin became unoccupied. So we can end up with another curve. It looks like this. And this would represent a PO2 of roughly 100 for argument's sake, i.e. the arterial side of the circulation. And this would be a CO2 of approximately 40, or the venous side of the circulation. And so the Haldane effect shifts these curves. Between here and here is a visual representation of the Haldane effect. That's another way to explain it. But what it does mean is that if we have a curve that shifts in the presence of changing oxygen tension, we have a changing ability to hold onto CO2. i.e., there's a greater capacity to hold CO2 in a low oxygen environment, because this curve shifted upwards, than there is in a high oxygen environment, because this curve is lower. So let's look at the situation that occurs when we transition from an arterial circulation to a venous circulation in the carbon dioxide content. Again, we're going to talk about the tissues. And the tissues would start at a low carbon dioxide tension. For a given carbon dioxide tension, for example, in the arterial circulation, we would start at this point. If we do nothing more than remove carbon dioxide, what we're going to do, remove oxygen, what we're going to do is result in an increase in the amount of CO2 totally in the blood. So what we end up doing here is just simply by removing oxygen, simply by the Haldane effect, we're going to increase the amount of carbon dioxide that can be loaded into the blood. And that's extraordinary. The other thing we can do is talk about a low oxygen situation in the tissues, such that in the situation of a little bit higher PCO2 existing in the tissues, rather than have a line that stops here, we're in a lower oxygen situation. in the tissues, so that we're no longer in the lung, we're on this curve, on the venous side of the circulation. So in the tissues, where there is a relatively higher PCO2 environment and we have a relatively lower oxygen environment, we in fact end up with this amount of total carbon dioxide bound. And so the Haldane effect produces a much larger capacity to move carbon dioxide just by dropping oxygen off hemoglobin. And that speaks to this large reservoir that exists, that we constructed here, that holds 93% of our capacity. So just through the Haldane effect alone, and that's why this thing is important, is that it allows the shift in the curves and allows this much extra carriage of oxygen. So that's a visualization of the Haldane effect. And shifting from one curve to the next, because of Haldane in different oxygen environments, and shifting from a given curve in different tissue CO2 tensions is another way that these questions are asked. So we'll get on to asking a question. In what forms is carbon dioxide transported in the blood? Carbon dioxide, much like oxygen, is transported both dissolved in blood and bound. Dissolved proportions are related purely to the PCO2 and equate to approximately 7% of the carriage of carbon dioxide in blood. The bound portions are related to transformation in red cells to bicarbonate ions using carbonic anhydrase, which are then channeled out into the plasma and rely on a chloride shift for that. It's also attached to hemoglobin in the unoxygenated state and does so according to the Haldane effect. It also exists as carbon dioxide converted to bicarbonate and attached, the resulting hydrogen ions are attached to the hemoglobin. Please draw the carbon dioxide dissociation curve for normal arterial blood. Carbon dioxide dissociation curve relates total CO2, which is a mass term, to the PCO2 or partial pressure of oxygen. It describes a near linear portion of the curve between 40 and 50 millimeters mercury. That describes, for arterial blood, the narrow physiological range between 40 and 45 millimeters mercury of carbon dioxide. Where will the curve lie for venous blood and why? Venous blood describes a different curve to arterial blood. Arterial blood, or blood which has a PO2 of around about 100, would describe this curve. If we deoxygenate that blood and evoke the Haldane effect, we shift the curve in this way. So this will be the venous side of the circulation with a PO2 of 40. And the Haldane effect is an increased ability of hemoglobin to accept carbon dioxide in its deoxygenated form. And that results in a shift of the curve here. what that really means is that at a given tissue PCO2, the carbon dioxide carrying capacity of the blood is increased by this amount. And that's it. Thank you very much for this series of lectures. I hope it has been helpful. It's a unique way to try and give this series and convey a lot of information. I hope it has allowed you to pull together some of your reading into a comprehensible story that relates a lot of the different questions to a single body of knowledge. A lot of these things in respiratory and cardiovascular seem like they're light years apart and totally different diagrams. If you take anything away from this, I would like you to try and take away the point that they're all related and there's a journey of knowledge. There's a few chapters that relate to each other that explain what has gone on. And I hope that we've been able to show you that on the way through. Thank you very much. Good luck with exams.